Okay, sir, it is audible.
Yes, audible.
Okay, sir.
Yeah, let me start. Basic introduce yourself, then we'll be discuss.
Okay, thank you, sir. Myself, Krishnadipti Singh. I am 32-year-old. I came from Tamluk hometown, which is under Purbo Medinipur district, West Bengal state. I have completed my graduation in chemistry from Calcutta University in 2012. I have completed my post graduation in chemistry from IIT-GUWAHATI in 2014. I have completed my PhD from CSIR CSMCRI in 2021. Some of my achievements are I got West Bengal Merit Cum Scholarship in 2007. I have qualified JAM exam, which gives me opportunity for MSc in IIT Guwahati. I have qualified a GATE exam and CSIR GRF, which gives me opportunity for PhD in CSIR CSMCRI. Some of my hobbies are in leisure time, reading and listening of some story books, particularly in detective stories and scientific innovations. These are my hobbies.
Yes, what the meaning of your name? Explain briefly.
Sir, my name Krishnadipti Singh. Its inner meaning is ray from valuable black body, particularly from diamond.
Yes, good. Your hobby is playing cricket, yeah, storage. Yes. So how to use
Yes,
it?
sir.
Describe your hobby. How to use?
How to use my hobbies?
Yes, in a day, week, month.
Sir, actually, leisure time I have playing cricket for developing good fitness and mental health, which also gives me fresh energy.
Yes. Next, move on laboratory sections and native place. First of all, your native place. Your native place is
My native place,
Modnipur, West Bengal.
Tamluk town, which is under Purbo Medinipur district, West Bengal state.
Yes, Modnipur,
Uh,
West Bengal.
so
Yes.
native,
Yeah, describe your
yes, sir.
native place. How do you feel that?
Okay, sir. My native place is Tamluk hometown. It has some importance. It is situated in the bank river of Rupnarayan. Lots of people visited here for picnic in holidays. Here, one famous temple named Ma Borgovima temple, uh, which was considered as 51 Shaktipeeth of Ma Durga. It is considered as a 1115-year-old Ma Kali temple. So lots of people came here, this temple, and one famous railway station in Tamluk, which is dedicated for one lady revolutionarist named Matangini Hajra. He actively take part in Indian independence movement, uh, in particularly in 1942 in Quiet India movement he joined, and at that time he was died. So this is the one of the historical importance of this area.
Yes, what are the famous place in your district and state? Explain. Only 10 minutes. Yes, explain.
Sir, famous play, actually lots of people here playing cricket, short hand cricket especially, and also play some short football tournament to These type of two games are actually played in my hometown. Only short football tournament and cricket tournament
Yes, no problem.
during some season.
Yes. Next, move on. The laboratory section. What is lab? What the role of laboratory in your life? Explain briefly.
Sir, chemistry, particularly chemistry subject, which is mainly associated with lab section. From beginning BSc, here we habituated with lab sections for detection of some important element and detection of some important element and functional group. Then in higher section, uh, we are calculating some unknown concentration of sample by using titrations and other potentiometric method. Then in MSc portion, where we have it, we use some instrumental technique to understand the some property of molecules and their electronic interactions, and using some instrumental knowledge to identify the structure. Then in my PhD section, I actually we are habituated with these instrument sections for making some new molecules and advanced use of some other instrument technique like which is specially gives informations about some molecules and their uses. And also we in my PhD I utilize all these sections for application in society, chemistry application in society. So these are the lab sections I used in my education career.
Yes, what is functional group? How to detect it functional group in a lab?
So functional groups detected in lab by using some chemical technique. Like if I take one example, if aldehyde group present, then we used 2,4-DNP test for identify the particular group. And also if we want to confirm that particular group, then in higher section we used instrumental technique like IR, which actually gives the particularly frequency wave number values that corresponding that group confirmly present in that sample.
Yes, what is analytical chemistry? Analytical chemistry is the 5 part: qualitative, quantitative, volumetric, gravimetric, instrumental analysis. Explain one by one.
Sir, analytical chemistry parts are qualitative analysis. It gives basically the elementary idea about the molecule, means that some type of that elements or functional group that are present in the sample, which are confirmed by by this qualitative analysis. Then in quantitative analysis, here we used some techniques to know the unknown concentration amount of sample and calculating the percentage of that particular species present in the sample. So the qualitative analytical part sections, there are lots of technique. Like among them, one is gravimetric. Gravimetric analytical, that is one of qualitative part. Here actually we measure the amount of here technique used is the precipitation of that particular element. From that precipitate, we calculate the amount of species that present in the sample. This is the gravimetric technique. And also other techniques are like using some spectrometer by that is using the Lambert-Weir law. that is calorimetric method. Here we use the UV with the help of UV intensity. We measure the concentration of unknown sample by looking their UV visible curve, and also other titration based methods also present here, like example, that is acid based titration, where we using one primary standard solutions and secondary standard solution, that is we have to determine by using that acid based titration. We can measure the amount of unknown amount of sample present in it. And another part of titration is redox. So it is basically the redox reactions, means oxidation reduction part. Here also we used primary standard and secondary standard. Then to calculate the unknown strength of the sample. And also analytical part, beyond them, we use some instrumental techniques to measure the unknown concentration of sample by using HPLC and GC. It also measures the purity of the sample.
Next, move on MSc project.
Okay,
What
sir.
main application of your MSc project?
Sir, my MSc project topic is synthesis of triazole-based unnatural nucleosides. So main application is that synthesis of triazole has some important applications, like in the field of, and it can be used as a anti-cancer drug, or it has some property like antiviral and antimicrobial property, and it also used for some cell imaging, for sensing some DNA molecules. For these type of important applications, so researchers are interested for synthesis of this triazole based moiety.
Yes, what the future of your topic? What are the futures?
Sir, future of topic is that here we basically synthesized this triazole moiety by using click reactions between alkyne and azides. Here we varied azide moiety by taking sugar unit as a fixed. So here one azide part that came from anthracene, that gives the result of that particular triazole, which is highly fluorescent active. So it can be used as a cell imaging, particularly in case of so as it is highly fluorescent active. So after incorporation of DNA unit in that particular moiety, it can be sensed as a particularly some sensing for biological cell imaging. And another part is that this type of triazole moiety, we found that from some research article, that it can be used as anti-cancer and antiviral and antimicrobial type of applications. So it can be used for that also.
Yes, any drawback, limitation of your MSc project?
Sir, main drawbacks are some heterocyclic azides, which reaction with this sugar moiety alkene actually results poor yield of triazole. And another drawback is that in case of aliphatic azide, it also gives the lower yield of triazole unit that we have synthesized. So these are the main drawbacks.
Next, move on your PhD topic. Yes, photocatalysis and heterogeneity and different organic redox reaction. What the application of this topic Explain briefly.
The main application is that here we used heterogeneous photocatalysts like CDS TiO2 and CDSE composition of some metal impregnated photocatalyst for particularly application of mainly in my case I used these photocatalyst for some valuable organic transformations for synthesis of some N-hydroxamide esters, active esters, which is important intermediate for synthesis of some amide type of drug molecules. Particularly two amide type of drug molecule I have synthesized. One is moclobamide, which was used for hypertension, and another is idropide, which was used for nausea and headache. And another important part of this photocatalyst is that after reaction, we can reuse this heterogeneous photocatalyst, which also uh, one which also have one important part that it not generate any hazardous byproduct during reactions. And another point is that this type of heterogeneous photocatalysts are used for production of hydrogen from water splitting. As we know that hydrogen is one of the green and clean economical energy source, which can be used as a fuel in future. So particularly these type of applications are important in my PhD topic. And also future of this project is that these heterogeneous photocatalysts are used for making solar cell, and these type of heterogeneous photocatalysts are used for degradation of dye molecules, which are the one of the important byproduct that are released from textile industry.
What is photocatalysis reactions? How many types of photocatalysis reaction?
So photocatalysis reaction is that technique that used uh, catalyst, photocatalyst means some catalyst which are convert the photonic energy into either chemical or electric energy. There are two types of photocatalysis. One is homogeneous and another is heterogeneous. In case of homogeneous catalysis, there catalyst and a reaction mixture present in same phase. But in heterogeneous catalysis, here catalyst and reaction mixture are present in different phase. If I take one example, that iridium bicomplex, that is one of the homogeneous photocatalyst, which were mostly used uh, as a photocatalyst for homogeneous cases. And heterogeneous catalyst, one example, that is titanium dioxide.
Yes, how many types of heterogeneous reaction? Now, what the difference between heterogeneous and homogeneous reactions?
Sir, type of heterogeneous, that is basically that are solid, actually catalyst actually present in solid phase, and uh, reaction mixture that present in liquid phase. So sorry, question is that how many type of heterogeneous photocatalyst and
What the difference between heterogeneous and homogeneous catalysis?
homogeneous. So heterogeneous and homogeneous catalysis difference is that in case of homogeneous catalysis, here we cannot reuse that catalyst after the reactions, and in case of heterogeneous catalysis, we can reuse that heterogeneous photocatalyst after the reactions and reuse several times. And also No byproduct formation occurs in case of heterogeneous photocatalysis, so which is environmentally friendly.
What is redox reaction and how many types of redox reaction?
So redox reaction, that is continuously uh, oxidation and reduction reactions occur simultaneously. Type of redox reactions, that is intramolecular redox reactions and intermolecular redox reactions. In case of intermolecular redox reactions, here two different species, that one is oxidized and another is reduced. If I take one example, that is ferric chloride, that is Fe is in +3 oxidation state, which reactions with uh, nitrate, which gives where copper is in uh, +1 state. After the reactions, that Fe3 plus, that reduced to form Fe2 plus, that is ferric nitrate, and copper from cupric chloride. So this is one type of intermolecular reactions. And intramolecular reactions, uh, here actually it is happened in same molecule, that is that some intermediate stage actually, that uh, intermediate oxidation state of that particular species, that is simultaneously oxidized and reduced. If I want take one example, that hydrogen peroxide, which gives oxygen and water, so simultaneous oxidation and reduction occurs here in from H2O2 to form that O2 and water.
Any drawback or limitation of this topic?
My PhD topic, main drawback is that in some cases, that photo recombination of electron hole pair occurs during the reactions. So in that time, the progress of the reactions become very slow and uh, results lower yield. So particularly that synthesis of that metal, in case of some metal impregnated heterogeneous photocatalyst, here during some reuse that particular catalyst, some metal leaching occurs. So reuse of this catalyst, that makes actually challenge. And another point is that uh, during the reaction, sometimes photo bleaching occurs. So catalyst particularly lose their activity during the reaction. And also basically lot of disadvantage that mainly occurs with CDS. Here that particularly that catalyst actually associated with some drawbacks. During the reuse, we particularly faced the problem with CDS particular. And another drawbacks are that aliphatic and heterocyclic compounds actually result lower yield in case of heterogeneous photocatalysis, that I have observed in my PhD.
What the main function of redox reaction in your topic?
Sir, main function of redox reaction in my topic is that here I used photocatalyst, heterogeneous photocatalyst. Actually, if we look its structure, we found that there is a valence band and conduction band in my heterogeneous photocatalyst. So in presence of light, electron goes from valence band to conduction band. Here formation of hole and electron pair take places. Hole pair take places in valence band, and electron pair that is present in conduction band. So that electron pair that is used for reduction and that whole actually facilitate the oxidation process. So we consider the whole unit as a redox reaction.
Yes, a redox reaction like to inner sphere and outer sphere mechanism also a redox reaction. Yes. What the difference of each one? Inner sphere and outer sphere?
Okay, that particularly in case of some inorganic molecules, inner sphere means that reaction actually that some bridging ligand that present. Sorry, this is outer sphere. Some bridging, some bridging type of ligands actually facilitate the outer sphere mechanism, which actually bridged between them and facilitate the electron transfer from one species to another species. And in case of outer sphere, that is particularly inner type of that is higher oxidation state metals, where actually chance of that electron transfer is not possible for via bridging ligand. Not only that, some pi acceptor type of ligand present, which also making the molecule as a more electron particularly in these cases. So outer sphere reaction take places via outer sphere mechanism. Here also oxidation and reduction take places simultaneously. Yeah, if we take one example, that bipyridine complex of ferrocene and ruthenium complex. That bipyridine here actually that electron transfer take places where one is oxidized and another is reduced. Means if iron present in +3 oxidation state, that only gives Fe2 plus, and ruthenium, if it is present in ruthenium 2 plus, then it will convert to ruthenium 3 plus. Here actually electron transfer not take places from that electron transfer places from outer surface. So here no bridging ligand actually present. Sir, is this answer is right or I?
Yes, yes. One electron transfer and two electron transfer. Simple answer is yes, no? Simple basic point. Inner sphere, outer sphere, inner sphere. Yes, no?
Sir, in both cases actually oxidation state changes. But one difference is that here in case of inner sphere inner sphere mechanism, actually electron transfer via some bridging ligand, it will facilitate. But in case of outer sphere, that type of reaction, that is simple redox reactions, only electron transfer take place.
Yes. So many examples now. One electron transfer, outer sphere mechanism. Inner sphere, so many, yes. Ligands. What the difference between one electron transfer and two electron transfer in a redox reaction?
Sir, hello.
Yes,
Sir, it is audible.
yes. Whatever.
Okay, sir.
Yes, I'm asking regarding to one. What the difference between one electron transfer and two electron transfer? What the difference?
It is audible.
Yes, audible.
Okay, okay. Sir, between one electron transfer and two electron transfer process redox reaction, that is, there is in case of one electron transfer redox reaction, so only one electron will be transferred during the reaction, and in case of two electron transfer process, that two electron will be transferred during this redox reaction. This is simple explanation of that part. Means if we take one example, that in case of galvanic cell that made by zinc and copper, here in the whole process, two electron transfer process takes place in the whole reaction. But in case of hydrogen, if we consider some hydrogen electrode that have H22H plus, there actually one electron transfer process takes place.
Yes, good. What the future of your topic? PhD topic, future. Any 5-10 point explain briefly.
Sir, future of my PhD topic is that here we used heterogeneous photocatalyst, CDS, CiO2, and their composition, which will be used for making solar cells and some other electronic devices. And also it was used for production of hydrogen from water splitting, because hydrogen is considered as one of the green economical energy source, which used as a fuel in future, which also eliminates environmental pollution. That is one of the important feature of this project. And another point is that this type of photocatalysts are useful for degradation of uh, dye, dye molecules, which are uh, that dye molecules that are byproducts from in textile industry. So these dye are very carcinogenic in nature. These dyes are degraded by using photocatalyst in presence of visible light by using heterogeneous photocatalyst. So these are the future of uh, my PhD topic.
Any presently example of your MSc project topic? Any presently exam that people are using in your MSc project topics, materials?
Actually, that particular professor I have worked MSc project, that triazole based moieties they are used for their uh, DNA, used for DNA sensing for, and also used for making some cell image. Actually, because this type of moiety shows photoluminescence property that they are studied. So these type of applications actually are used by that group. And I also found some literature study that this type of triazole unit actually used uh, for making some anti-cancer drugs, and also it can be used for as a it is used for a antifungal and antiviral type of property. So lots of researchers are work in that particularly that field.
PhD topic, what the uses in our society presently?
Uh, in, in my PhD time, we have one coursework that is CSR 800. That in that coursework we have to use our uh, we have to use our knowledge for making society benefit. So in that case, uh, I used some seaweed-based photocatalyst by making with some heterogeneous uh, that is particularly some bismuth type of molecule by impregnation, making some photocatalysts particularly based on bismuth sulfide. These heterogeneous photocatalysts are used for dye degradation. So as we know that dyes are one of the byproducts from textile industry. These are carcinogenic in nature. So these type of dyes are degraded by using this photocatalyst. So this is one of the uh, important applications for society, because here actually we taking that bio-based seaweed that are cultivated in ocean region. So these type of seaweed actually the good source for sulfur. So by using that sulfur source, we making this type of heterogeneous photocatalysts that are useful for society.
Yes. What the uses of heterogeneous catalysis in your topic? Any point 4-5 point uses?
Main important part is that the heterogeneous photocatalyst that is reused after the reaction and giving more cycle uh, after during the reaction. So reuse is the one of the beneficial part. So after reuse, the catalyst is taking its activity. So and another important part is that this type of heterogeneous photocatalyst that not release any byproduct during the reaction. So it is not it is one of the eco-friendly and not create any problem in the environment. And another important point is that here we particularly use heterogeneous photocatalyst. So it used energy source as a visible light. So which type of reactions that basically we need energy that from by heating. So it saves energy and also economically effective because it also saves money. And after the reaction, we use the catalyst. That is one of the advantages.
What the relation between in your topic is photocatalysis, heterogeneous, and redox reaction? What the relation of these three topics in a
Relation, heterogeneous part that came from particularly catalyst. Here, actually, catalyst and the reaction mix are present in different phase. That if we consider one example, titanium oxide, and that is used for organic transformation for making N-hydroxamide ester.